Dear colleagues, my name is Anastasia Ilina. I am a research assistant of the Laboratory of Molecular Mechanisms of Aging at the St. Petersburg Institute of Bioregulation and Gerontology. I am a PhD student of the Department of General Pathology and Pathological Physiology at the St. Petersburg Institute of Experimental Medicine. Today I'd like to focus on the effect of short peptides on brain functioning under normal and neurodegenerative conditions. First of all, I'd like to emphasize on the potential neuroprotective peptides. The EDR peptide is an active component of the dietary supplement Cellutin. Let's talk about relationship between short peptide and dietary supplement. The drug cortexin was developed based on polypeptide complex obtained from the extract of the cerebral cortex of young calves. The dietary supplement Cellutin was developed based on a polypeptide complex obtained from the extract of the whole cerebral cortex with white matter. The pinolone peptide was synthesized based on analysis of amino acid composition of polypeptide complex from the extract of the cerebral cortex of young calves. The next peptide is kAT. This peptide is an active component of the dietary supplement Ventfort. Let's talk about relationship between this peptide and dietary supplement. The drugs Levinorm and dietary supplement Ventfort were developed based on a polypeptide complex obtained from the extract of the vessels of young calves. The Vasogen peptide was synthesized based on analysis of amino acid composition of polypeptide complex from the extract of the vessels of young calves. Moving on, I'd like to focus on the effect of short peptide on brain functioning under normal conditions. One of the fascinating research was devoted to the effect of EDR peptide on memory formation in honeybees. Let's talk about methods of this investigation. The objective was to evaluate the conditioning of the proboscis extension on olfactory stimulus. How bees were selected. The sensory and nutritional excitability of bees was assessed in the first step of the experiment before the training. The sensory excitability reflects into the presence of spontaneous reaction of the proboscis extension on the unshaped smell. The nutritional excitability reflects into the proboscis extension in response to the contact of the sucrose solution with the antennas. Bees were not used in the experiment if they had sensory excitability and or no food excitability. How peptides were injected. Dorsal injections of EDR peptide solution in a range of noted concentrations of saline were made in the bee stomachs for 30 minutes before training. What was retraining? A conditioned reflex of the proboscis extension was developed by a single combination of close smell and food reinforcement in bees fixed for wings. What was the conditioning? Bees were checked for a conditioned reaction to smell for noted time after training for short-term and long-term memory testing. The number of conditionally respondent bees were estimated as a percentage of the control level. All insects were divided into two subgroups in this study. The reflex conditional background deviated from the mean to the lower side in the first group that corresponds to the figure on the left. The control reflex conditional background deviated from the mean to a large direction in the second subgroup that corresponds to the figure on the right. The administration of the EGR peptide stimulated memory processes involved in maintenance of the conditioned reflex in bees with initially low food motivation. The administration of the EGR peptide decreased the number of bees exhibiting the conditioned reflex in memory with initially high food motivation. Thus, the EGR peptide modulated the memory formation process depending on the initial condition background. The next fascinating research was devoted to the effect of EGR peptide on condition of rhesus macaques. Let's talk about methods of this investigation. Healthy macaques were used in this study. The objective was to assess the effect of EGR peptide on learning and sustained attention of macaques in the presentation of informative signs in visual stimuli. What was the scheme of peptide injection? Learning and sustained attention of macaques without administration of EGR peptide were studied at the first step of the experiment. A year later, macaques were subjected to a course of EGR peptide sublingually per day, followed by experiment was repeated to assess learning of monkeys before and after treatment. Experimental design was following. On the first step, visual perceptual learning by operant conditioning was assessed. Let's click on the neutral image on the touch screen of the monitor and move it aside. On the second step, two learning tasks were performed. First was a choice of black visual stimuli, and second was a choice of stimuli with sharp angles. The monkey was considered trained if it has 75% of correct responses. On the third step, a new set of 30 visual stimuli containing 4 elements but with the same informative sign was presented. The first result was focused on learning of rhesus macaques. The administration of the EGR peptide contributed to a statistically significant reduction in the number of visual stimuli required to rhesus macaques to assimilate the principle of informative sign detection in the learning process. This indicates an acceleration of learning process of rhesus macaques under the influence of the EGR peptide in the paradigm of visual perceptual learning by operant conditioning. The next results are devoted to a successful completion time of rhesus macaques. The administration of the EGR peptide in rhesus macaques contributed to a statistically significant reduction of successful completion time during selection of informative signs among visual stimuli in training step with one element. Completion time in macaques injected to EGR peptide was reduced to 30% in the control test phase with 4 elements. This is further evidence supporting the acceleration of the learning process of rhesus macaques under the influence of the EGR peptide in the paradigm of visual perceptual learning by operant conditioning. Moving on, we can see the results focused on sustained attention of rhesus macaques in learning process. The administration of the EGR peptide in rhesus macaques contributed to increase the correlation between the number of concentration reactions, while correlation was low before the EGR peptide administration. This indicates the increased ability of macaques to focus on selected stimulus under the influence of the EGR peptide in the paradigm of visual perceptual learning by operant conditioning. Now let's turn to the effect of short peptides on brain functioning under neurodegenerative conditions. The first neurodegenerative condition we will discuss is Huntington's disease. Particularly, I'd like to focus on the neuroprotective effects of EGR and KID peptides in vitro model of this disease. Huntington's disease is a genetic progressive neurodegenerative disease that was firstly described by George Huntington in 1872. Generalized choric hyperkinesis, dementia, and behavioral impairments are among the classical clinical triad characterized of HD. The neuromorphological features of HD are characterized by atrophy of striatum at the earliest stage of this disease, then degenerative process spread to other areas of the brain, thalamus, cerebellum, substantia nigra, and globus pallidus. Further neuronal death leads to atrophy of whole brain. GABAergic medium spiny neuron of striatum are the most affected in HD, where the concentration of gamma-aminobutyric acid is reduced. Let's talk about method of this investigation. The objective was to evaluate morphological changes of neuronal dendritic spines under influence of the EGR and KID peptides in HD model in vitro. The model of Huntington's disease was primary dissociated corticostriatal cultures of neurons taken from the brain of newborn mice of wild type and transgenic line with mutant gene of human huntingtin. The EGR and KID peptide were added to culture medium at the noted concentration. Immunocytochemical staining of corticostriatal cultures of neurons was carried out at 14 and 21 days of culturing to verify the marker of medium spiny neuron in striatum and neuronal marker. Microphotographs of neuronal cultures were taken by means of confocal microscopy, followed by automatically counting of spines. Now let's turn to the results. The EGR peptide at the 20 nanogram per milliliter concentration didn't affect the number of spines in primary dissociated corticostriatal cultures of neurons. The EGR peptide at the 200 nanograms per milliliter concentration contributed to increased density of spines to 35% compared to control neuronal culture without added EGR peptide. The KID peptide at the all concentration didn't affect the number of spines in primary dissociated corticostriatal cultures of neurons from wild type and transgenic mice. Thus, the EGR peptide has neuroprotective properties in vitro model of Huntington's disease. The EGR peptide contributed to the recovery of morphology and number of spines in primary dissociated corticostriatal cultures of neurons of young mice. The second neurodegenerative conditions we will discuss is Parkinson's disease. Particularly, I'd like to emphasize on the neuroprotective effects of EGR peptide in vivo model of this disease. Parkinson's disease is a slow-progressing neurodegenerative disease in the older people. This disease belongs to the degenerative disease of extrapyramidal motor system. Parkinson's disease is caused by progressive destruction and death of neurons producing the dopamine neurotransmitter, primarily in the substantia nigra, but also in other departments of the central nervous system. Insufficient production of dopamine leads to inhibitory effect of basal ganglia on the cerebral cortex in Parkinson's disease. The main symptoms of Parkinson's disease are muscle rigidity, hypokinesia, tremor, postural instability. Methods of this investigation were following. Model was fly's Drosophila melanogaster with dementia and Lewy body. The aim was to evaluate the locomotor activity and spital orientation of flies injected to the EGR peptide. How fly was selected? Fly's Drosophila melanogaster grew on standard dietary rhizine yeast medium. Testing was carried out on the third age larva and 5-day male imago. The EGR peptide was added to dietary rhizine yeast medium in the noted final concentration. Behavioral evaluation was following. Fly's behavior was registered in automated installation and recorded by digital camera. Digital recorder was analyzed by Locotrack software. Locomotor activity was evaluated by measuring the index of activity, riding frequency, duration, length, and speed of each drag. Visual spital orientation was evaluated by the shape of the trajectories of drags of the larva. Now let's turn to the results. Locomotor activity of controlled mutant larvae of Drosophila melanogaster without administration of the EGR peptide was decreased in comparison with imago. Locomotor activity of mutant larvae of Drosophila melanogaster with administration of the EGR peptide was increased, while this indicator wasn't changed in imago. The EGR peptide contributed to increased visual spital orientation in mutant imago and didn't affect in larvae of Drosophila melanogaster. Thus, the EGR peptide contributes to increased locomotor activity of flies Drosophila melanogaster with prominent symptoms of Parkinson's disease. The next results were devoted to the effect of the EGR peptide on gene expression in flies Drosophila melanogaster in Parkinson's disease model. Expression level of all 3 genes in normal are taken as 1. Mutant flies exhibit increased LIMG ROC genes expression and decreased PARC gene expression. The administration of the EGR peptide to mutant flies contributed to decreased expression of LIMG1 and ROC genes, which were increased in mutant fly without peptide injection. The EGR peptide contributed to increased expression of PARC gene, which was decreased in mutant flies without peptide injection. The EGR peptide didn't affect the gene expression in normal flies. Thus, the EGR peptide is likely to influence on locomotion and memory through decreased expression levels of LIMG1 and PARC genes, which play a significant role in the dynamic of actin filaments. In conclusion, it should be pointed out that the EGR peptide has neuroprotective effect on normal aging as well as in neurodegeneration. The EGR peptide and the complex dietary supplements based on the cattle vessels are appropriate to be used in order to prevent age-associated loss of cognition in normal aging and in neurodegenerative disease. That's all I have to discuss in the present lecture, so thank you for your attention.